Hello, people. Welcome to developmental biology. I know. I know. You've seen it all before. Let me see if my face looks better if I don't have this virtual background. I think it looks worse. No, it's worse. It's much worse. All right, let's bring the dog up anyway. Dog. Okay, let's say see your faces so we can all say hello. Pretend we're a class for a moment. Elijah, Angelique, Audra, Michelle, Esha. Hey, Esha. Christopher James still not smiling. That's okay. Anthony Estevez. Yusuf is shown. Hey, everybody, Yusuf is here. Now we can have a class. I was going to cancel class, but Yusuf showed.
I'm sorry, guys. Today's my birthday, by the way.
Oh, you said that a week ago.
Yeah, I said it was coming up.
Oh, jeez.
I
can't take this exam because my birthday's in a couple of weeks. What? You're the worst, dude. Okay, good news, people. We're going to rock and roll today. Today's the day. We are doing early mouse development. I know how you love this. I do anyway. Oops, I'm on the wrong slide. Stupor. Okay, I'm going to go over this, what we did Friday, really quick, just to catch us all up. Hang on a second. Go away. I'm just trying to set up my real estate here. Go away, you. All right. No, yeah. I think it would all be better if I was at work because I'd have a bigger screen. I'm just on this little laptop screen. Okay, come on, you. Struggling a little bit with my desktop here. Okay, so this is where we started last time, and it was a short one. Friday was a short lecture. Today's going to be a rock and roll lecture. So we started out with the mouse morula, about 8 cells. It starts to compact, and when it compacts, it ends up with cells that are all expressing CDX2 and OCT4. When it squeezes itself together, it ends up with inside and outside cells. Those inside cells are touching on all their surfaces. The outside cells have an apical surface that is not touching anything. It's free. That's what I meant to say. And so because of that, the inside ones start to produce only OCT4, and the outside ones produce only CDX2. And CDX2 will turn those outside cells into trophectoderm, and the OCT4 will become the embryo proper. Now, why is that happening? It's because of hippo signaling. So hippo signaling later will be important for determining how big organs get before they stop growing. But here are our inside cells. Oh, you know what? I don't think I uploaded. Oh, I'm sorry. Let me just stop for a second here. I didn't load the notes today. Let me do that real quick. So you guys can see the notes while I'm yakking away here. Exam 3. Okay, lecture 421, lecture 4, 2021 is up now, so you can look at that. All right, sorry about that. Back to work. Okay, so your inside cells are touching everywhere, and that causes Hippo to be released. Hippo is a kinase, and it phosphorylates warts, and warts then becomes an active kinase as well. It phosphorylates YAP, and YAP is a transcription factor that then gets targeted to be broken down by the proteasome. So in the absence of an open or a free side, when you're touching all over, you activate Hippo and you break down YAP. When you do have a free side open, that causes this AMAT to come off of this KEM complex, and Hippo goes onto it. So then Hippo is inactive, which means WTS is inactive. So YAP builds up, it binds to TEAD, and together they're a transcription factor, and they activate the transcription of CDX2. And as we all know, CDX2 blocks OCT4 transcription, and OCT4 blocks CDX2 transcription. So Hippo or no Hippo depends whether you're going to get only OCT4 or only CDX2 because they inhibit each other. So our trophectoderm cells then are going to be expressing OCT4. Okay, so here is a video that's going to talk about that. I think it's a pretty cool video, so I'm going to go ahead and show it. I don't always do this, but boom, ba boom, boom, ba boom, ba boom. Oops, wait, what? Hang on a second. You share this. Okay. So this is about stem cells, but the first part of it is about its development.
Cells are differentiated, meaning they have taken on specialized properties and functions and have lost the ability to generate cells of other types. Skin, muscle, blood, bone, and the nervous system are all made up of populations of differentiated cells. There is another type of cell, however, that remains less differentiated and retains its ability to give rise to other cell types. These cells are known as stem cells. Stem cells exhibit two characteristics not found in more differentiated cells. By definition, stem cells are able to divide and make copies of themselves indefinitely, a capacity known as self-renewal. This endless power of division is not limited to creating more stem cells, however. They can also give rise to a wide range of mature cell types. These unique characteristics make stem cells indispensable, both for replenishing the body's cells as they age and are lost, and for constructing the body itself from a tiny number of cells during development. At the very earliest stages of development, all cells are equally capable of giving rise to an entire organism. But after only a few rounds of cell division, the cells begin to lose this totipotency and to differentiate, sorting into groups that will either contribute to the placenta or to the embryo itself. In this 8-cell embryo, called a morula for its berry-like appearance, all cells express a pair of genes, CDX2 and OCT3 and 4. But as the cells of the morula go through their next round of division, CDX2 is downregulated in the innermost cells, while OCT3-4 remains expressed throughout. The segregation of these genes is maintained as the cells continue to divide, until after about 10 rounds of division, the embryo has become a hollow ball, known as a blastocyst. The outer layer of the blastocyst is made up entirely of cells that express CDX2. This trophectodermal layer will go on to become placenta. On the interior, OCT3-4 marks a small cluster of cells with phenomenal ability to differentiate.
Phenomenal.
This inner cell mass is made up cells with the developmental potential to give rise to all of the body's multi-cell lineages, an ability known as pluripotency. And indeed, every one of the cells in your own body can trace its ancestry back to these few dozen pluripotent stem cells.
Okay.
Even after the embryo has grown into an adult, many of the
All right. Now after that, it's less specific to this class, but you can see how that is basically what we said. Okay. So I will post if you want the link for this, it's in the PowerPoint, so you can get it that way, or you can. I will post it because I'm that kind of guy. And let me just update the PowerPoint as well. Okay. So, what will we talk about today? Oh, damn it. Okay. What will we talk about today? Good question. So what happens next? Okay, so now we've got a group of cells that are going to be trophectoderm expressing CDX2, which it kind of, it's a differentiation process. So it's kind of terminally differentiated in that it's never going to go back and become embryo again. So that's our first one. But the inner cells are expressing OCT4, and that keeps them more pluripotent. Okay, so at this point, the cells are all the same. Each one of them could become a complete embryo. But they're going to start to differ. And the way they differ is in these two transcription factors. So early on, they both have NANOG and GATA6. I thought it was GATA4, GATA6 at low levels. And then for some reason, although it's not quite clear, some start expressing more NANOG and some start expressing more GATA6. Well, more to the point, some start expressing more NANOG. And NANOG then causes the expression and release of FGF. Here comes our old friend FGF again. Always important here in early embryo development. So FGF starts to be released from these blue cells, and the yellow cells have got receptors for FGF. So here we're signaling from one cell to the other. And FGF is going to cause those GATA6 cells to become primitive endoderm. Primitive endoderm, which will be really important for early development here. So they segregate then, and you end up with your primitive endoderm on the outside. Now, this primitive endoderm is extraembryonic, but it's going to be, you will see later today, releasing factors that will cause differentiation in the ectoderm, which is what the blue is, right? Right now, it's just epiblast, which basically means ectoderm. They're all ectodermal right now. If you took them out and cultured them, they'd be skin. So anyway, this primitive endoderm then, which is extra embryonic, will be releasing factors that will pattern and differentiate the ectoderm. And here's again our trophectoderm, right? And this is going to be implanting. All right, let me make sure I didn't forget something there. Okay, so now at this point, the embryo has got a polarity, right? You can see this is different from that. So we've got a top and a bottom here. The top we're going to call the embryonic pole, because that's where the trophectoderm is going to invade the uterus and set up the placenta. So that's the embryonic pole. And then the other pole, because it's on the other side, is the aembryonic pole. See, the embryo isn't even here. We got this big blastocele in the middle and a bunch of trophectoderm, but there's no embryo down here. The embryo's up here. So this is aembryonic and this is embryonic. This polarity is not due to anything maternally laid down. Remember how important that was in Xenopus. Animal pole versus vegetal pole was by embryonic or the maternally laid messenger RNAs. Here, you don't get any of that. Right from the one cell stage, you're getting zygotic transcription. So again, just to review, we said in both the xenopus and zebrafish, you get no transcription from the embryonic nuclei until a certain stage. You get a certain number of cells, and then it starts. Here with the mouse, right from the get-go, you're getting transcription from the mouse nucleus. So this difference then between the top and the bottom is not set up by the maternal, but by the events that we just talked about. And then again, too, by the 2-cell stage, transcription really ramps up, and that's where all these changes start to happen. Okay, polar trophectoderm then, which is up at this pole, is going to implant into the uterus, and the proximal end, this is the proximal end, will becomes the placenta. Okay. So now, and this is where we're headed, right? So this is kind of the next step. Of course, it makes its way to that step, but we've got a number of structures here that are going to be important. I'm going to just define them first, and then we will show what they are and what they do as we go through this early pre-gastrulation stage. So out here, this was trophectoderm, and it's now become the ectoplacental cone. So it's developing the placenta out here. Now this is cut away because you still have trophectoderm going all around this and a blastocele in this area, but they cut it away to make it easier to see what's going on inside. Now up here, we've got extra embryonic ectoderm. So these were cells that were part of that, that epiblast, the ectoderm at the top that have divided, but they're going to be extra embryonic. They're going to play a really important role in patterning, but they're not going to be part of the embryo in the end. And these cells proliferate, they divide, and they force the other cells downward, and that's how we get this cup shape. So this is embryo proper here. So we've got ectodermal cells, and this is where the primitive streak will start. On the outside of that, encasing the whole thing, is that visceral endoderm I talked about. Those are the nanog expressing cells. Let me make sure I didn't get that wrong. Nope, they're the GATA6 expressing cells. Sorry about that. Okay, so this is your primitive endoderm. No, I'm sorry, visceral endoderm. Visceral endoderm, it's right there. Now, as we go through this, you'll see that this is going to be the anterior side, and this is the posterior side. If you remember from chapter 3, the mouse is inside out. So the dorsal side is on the inside, and the ventral side is on the outside. Of course, right now we've only got one layer of cells, so there's really no dorsal or ventral, but this will be the dorsal, and you'll see that gastrulation will come through into the outside. In the middle, we've got our proamniotic cavity, which will become the amniotic sac when this thing turns. And that's what we got here. So what's going on? What the hell is going on? Okay. So the polar trophectoderm then proliferates to form this extra ectoplacental cone. The extra embryonic ectoderm pushes this down, and so it starts as a flat thing, but then it becomes a cup because it's being pushed down. Okay, so I've kind of explained all this already. Primitive endoderm surrounds it all. Okay, so now we're going to talk about what happens at this stage. Once you're at that cup, we're kind of like late blastula now, so getting ready to gastrulate. So what happens now is the primitive endoderm here, right? The most distal part of that primitive endoderm then is called distal, not primitive, I'm sorry, I keep saying that, visceral endoderm. Distal visceral endoderm. So this would be proximal, closest to the mother, and distal is furthest away. So this visceral endoderm here starts to produce and release lefty 1. Okay, lefty is a nodal inhibitor. We've talked about lefty before, I believe. So these start to produce lefty. and lefty blocks nodals. And the DVE is also going to start releasing other blockers, things to block Wnt and BMP. We'll talk about them in a second. Now this ectoderm all around here is starting to release now nodals. So here, these red lines here are nodals. So this is being released everywhere, and it's affecting the visceral endoderm. And the nodals then are going to convert this DVE into the AVE, anterior visceral ectoderm. So it started out distal just because it's the furthest part away. Now it's going to start moving, and wherever it moves, that will be the anterior side of the cell because it's going to be specifying anterior. So your nodals then are coming out of the embryo proper, the ectoderm here, and it converts that DVE into the AVE. And the AVE is going to release, so it's going to slide up to one side. Now, which side it goes in, if you're looking at this thing in 3 dimensions, it's kind of random. There's really no signal there to make it go up one side versus the other because it starts right at the bottom. Once it becomes AVE, it slides up to one side. So now we've got it slid up on the left over here. So that's going to be our anterior position. And it's going to specify anterior structures, specifically brain. Okay, this is going to be releasing Dickoff, Cerberus, and Lefty. And they block, as you know, Wnt and BMP and nodals, right? So now we've got all of those blocked. Now, if you remember back again to our Xenopus embryo, where you had everything blocked, you end up with anterior structures. Same thing here. Okay, we're blocking we're blocking nodals, Wnt, and BMP, and so this ends up being anterior. Here's an interesting fact. I looked this up today. I'm like, Dickoff. I mean, when I look at that word, it says dickhead, right? And I think I said something about that earlier on. I'm like, why did they name it that? I looked it up. The internet, who knew? Everything's out there. Dick is actually a German word for thick. So it means thick head. For some reason with this, with the mutation in this, they got thick-headed embryos or thick-headed, what is the fly? I think it was found in the fly first, the Drosophila. But anyway, just so I can stop using the word penis before head, it just means thick head. Okay, so these are being released here. And that's going to specify that as anterior. Check my notes real quick to make sure I didn't, because I feel like I'm going really fast. So I want to make sure I didn't forget something. So this visceral endoderm then is kind of like the hypoblast in chick. So if we go back to slides 9 and 10, you remember we've got VEG1 and Wnt inducing these cells to produce nodals. So now we've got nodals and Wnt, and then we've got the hypoblast producing Cerberus to block it, right? So now we go up to mouse. And we've got, again, BMP, which is, you know, a TGF-beta-like signaling molecule. And we've got Wnt being produced too. We'll see that in a second. And this AV is blocking that. So it's kind of similar model. It's almost like they evolved from a common ancestor or something. Okay, so specifies the underlying epiblast as ectoderm. And just to emphasize this thing is going to move up to the side, but this BMP is going to stop it from moving too far. It's going to stop it right there. So now we've got two different sides to this embryo, and that's going to set up our anterior posterior axis. Anterior here, posterior sliding down and up around the other side. And now we're going to get the primitive streak formation. So this AVE over here is releasing those blockers. And what's going to start the primitive streak is kind of similar to check. We've got Wnt. And Wnt is being released from the ectoderm here onto the visceral endoderm. And it's inducing the visceral endoderm to produce Wnt and release it there. So we've got a high concentration of Wnt here. And Wnt is also going to be upregulating nodal, similar to check. And so we've got Wnt and nodal all building up over here, nothing over here because of the AVE. And that's going to start your primitive streak, which is going to come down this way from posterior to anterior. Okay. You good with that? Oops, damn it. Questions, comments? Okay, so as you notice, how often do I say the word nodal here? Nodal, nodal, nodal. Nodal is so important. It comes in so often here in early embryo development. And if something's important to life, then pretty much is going to be closely controlled, right? Because if something goes wrong and it goes out of control, embryo dies and then those genes don't get passed on, right? Whatever mutation caused nodal to be out of control will ruin the embryo and it won't make it. So there's a lot of different controls on nodal because it's so important in early embryo patterning. Okay, so we've got 4 different levels of control of nodal. Hang on a second. 4 different levels of control. First level is that when nodal is released, we didn't talk about this before, but when nodal is released, it's not an active enzyme, it's a proenzyme, or active hormone, it's a prohormone. So its normal primary structure causes it to have a domain that laps around it, so it's not going to bind to any receptors yet. That needs to be cut off by a protease before it becomes active. So you've got a protease there called proprotein convertase that needs to be in the vicinity of the cells that are going to respond to it. So, you know, that's another level of control. Do these cells have the convertase or not? If they don't have the convertase, they won't respond to nodal. So that's level 1, actually activating the enzyme, the hormone itself. Level 2 is then these blockers we keep talking about. Cerberus 1 and Lefty 1, 2 bind to nodal extracellularly and block it. Lefty also binds to the receptors and blocks the receptors. So this is going to block nodal activity. This is what's coming from the AVE and blocking it from starting a second primitive streak. Third level of control is level of transcription. I would call this the first, but your book calls it the third. And so we see this all the time where nodal increases the transcription and release of nodal, okay? So you get this positive feedback loop, and we'll see this especially at the proximal end of the embryo. Nodal being released by the ectodermal, the extra embryonic ectodermal tissue will cause nodal to be released by the embryo, which then makes this feedback loop. Negative, nodal from the epiblast produces the AVE, which then induces the production of nodal blockers, Lefty 1 and Cerberus, okay? So you've got this at the level of transcription, then you've got a couple different ways of controlling that. And then the third level, something we haven't talked about yet, I don't know how much you know, but if you had genetics, you may have heard about this, but micro mRNAs. Micro mRNAs are small RNAs that are antisense to the signal, and so they bind to the messenger RNA of nodal and break it down. So if it's to repress production of nodal, a cell could upregulate the microRNA that's complementary to nodal itself. So let's look a little closer at how that works. Okay, so here's microRNAs. So I described this in the notes too, but so microRNAs are transcribed as kind of a longer RNA, maybe 100 base pairs or so. And internal to the structure of that 100 RNAs are a couple of reverse repeats, kind of. So they end up being complementary to each other. So they form these hairpin structures. These are common in DNA and RNA because they kind of form a handle that can be grabbed. So inside the sequence here, this red stretch here is complementary to this one, so they bind together and they form this hairpin loop and another hairpin loop over here. So that stands out to this guy, dicer. Dicer is an enzyme that will come along and cut out the extra pieces of RNA and release only the sequence that is going to be complementary to the mRNA that's going to be silenced. So this is only about 22 bases now, and that will be grabbed by some other proteins, and they'll make this RISC complex. So the RISC complex then stands for RNA-induced silencing protein complex. RNA-induced silencing protein complex. So this RISC complex then acts kind of like CRISPR and Cas9, right? Remember, that was where it had an internal sequence that it then used to go out and find things with. So it's the same thing here. This guy's got an internal sequence, and whenever a messenger RNA for the targeted protein comes out, the RISC complex grabs it by that complementary base system and then chops it up into little pieces. So this is another mechanism of inhibiting the translation of a protein within the cell. So this, and again, another way that nodal will be controlled. Controlled by form mechanism. All right, I'm just looking over my notes real quick to make sure I didn't forget anything. You know, it took me all morning to put this lecture together, and I went through it in like 20 minutes. Okay, I think that's it. Let me just go over it one more time. So at the 8-cell stage, we're expressing CDX2 and OCT4, and then we get the hippo signaling complex that makes this only OCT4 and this CDX2. CDX2 then becomes trophectoderm, and the OCT4 expressing cells then start to express NANOG and GATA6. Some cells will express more NANOG than the other cells, and then they start producing FGF. The other cells then have FGF receptors, and that causes them to become more black or white here, more NANOG and no GATA6 and GATA6 and no NANOG. And then they kind of move past each other and they rearrange until GATA6 is on the outside, and GATA6 then becomes that visceral endoderm. So now you've got embryo proper here, visceral endoderm, and trophectoderm all the way around. This then will implant, and when it implants, the trophectoderm here becomes the ectoplacental cone, starting that placental process. You get extra embryonic ectoderm here that forces the actual embryonic ectoderm down and forms this cup-like shape, visceral endoderm all along the outside of it. Now that visceral endoderm then, the most distal part of it becomes the DVE, and it's DVE because it's releasing the lefty-1, which is a nodal inhibitor. And you've got nodals being released from the ectoderm all around this embryo, which is going to convert this DVE into the AVE, and it's going to slide up one side. And this BMP signaling is going to stop it from going up the whole both sides. It's only going to come up one side, and only so far. And this is now going to release all of our inhibitors, Lefty, Cerberus, and Dickoff. And I still stumble on that Dickoff. I feel like I'm going to get, reported to Title IX for using the name of this stupid protein. Okay, so anyway, you're inhibiting all of that, and then that makes this anterior and that's posterior. So this AV is what's specifying the anterior posterior axis. And then as this advances on the posterior side, you get Wnt being released by some cells opposite the AVE, and that acts on the visceral endoderm to cause it to produce Wnt, stimulating itself, and we're also getting nodals released, and then our primitive streak will start here. Now, I'd like to do the primitive streak, but I want to keep you guys on the edge of your seat because we will cover that on Wednesday. And then just to review this, we've got several different levels that nodal can be controlled at. One is at the convertase step, the enzyme being proteolyzed to release the active form of it. Our hormone blockers, transcriptional regulation, and then microRNA regulation. Okay, well, we've rocked through that pretty quickly. I was thinking, I didn't know if I was going to get through all those notes in one day. And here we are, it's like 22, and I've covered everything I needed to cover. How about we do a quick quiz? Ooh, there's an idea.
No.
Oh, Yusuf, this is your birthday quiz. This is your birthday quiz. Happy birthday. Okay.
Thank you.
How old are you?
22.
22.
Yeah, kind of old.
22. I wish I was that old. I would kill to be 22. No, I wouldn't kill. I would like to be 22 again. Okay, so here we are. Shit, son of a bitch. Turn editing on. That was a quote from a movie, by the way. Shit, son of a bitch. That was from Stripes, which is a much worse movie than I remembered it being. Show. Okay, so there. Now you've got a practice quiz up. Practice quiz 2. So why don't you guys go ahead and do that, and between now and Wednesday, and we will discuss the answers on Wednesday. Okay? We all good?
Sounds good.
Okay, thank you, Yusuf. You're the only person who talks to me. Alex, where the hell are you? Alex?
Yeah.
There she is. Okay. Everyone else okay? See, I worry about you people. I see blank screen everywhere, and I think you could be dead for all I know. I hope you're not, though. I like you guys. Okay, we all good? Do we have?
Yeah, we're good.
You don't have to send me the answers. You can submit your answers, and for the multiple choice, you'll get your answer back. There's an essay question which you won't get it back for, but it's a good question. Okay, well, I'm going to let you people go and enjoy the rest of your day, unless you've got a question or a comment. All right, I'll see you Wednesday.